Good afternoon, and thank you all for attending today's webinar sponsored by the AII Assembly on Allergy, Immunology, and Inflammation, AII. My name is Tetsuro Maeda, and I am an assistant professor, Division of Pulmonary Allergy and Critical Chemistry at University of Alabama at Birmingham. I'll be moderating today's webinar entitled AII Research Abstract Webinar Presentations, Part 1. As you can see, there's part 2 also this Thursday starting at 1 p.m. EST. So please also check that out. This webinar will include presentations by 5 abstract award winners at ATS International Conference 2024, and a discussion joined by our expert panelist, Dr. Anne Sperling, PhD, Professor, Division of Pulmonary and Critical Care Medicine at University of Virginia, and Dr. Megan Ballinger, PhD, Associate Professor, Division of Pulmonary Critical Care and Sleep Medicine at the Ohio State University. So this is a little special format for the webinar, but we asked that each presenter speaks up to 8 minutes with PowerPoint presentation. And then we'll have approximately 2-3 minutes after each speaker for comments and questions. For our audience, please use the Zoom chat function to enter your questions at any time. The session will be recorded and posted on the ATA website. A recording link will be sent to all registrants. All right, so the time is a little tight, so without further delay, we'll start with a presentation by Dr. William Daccarett-Bajanini, MD, at Johns Hopkins University. His abstract title is Decitabine Affects Lung Monocyte Macrophage Dynamics and Phenotype in Acute Lung Graft Rejection. So please go ahead and share your screen and start presentation.
Can you guys see my screen?
Yes.
Okay. Well, good afternoon, everybody. Thank you for this invitation and for being interested in our research here. I'm a current postdoc at Johns Hopkins University, and I work in lung transplantation research. And today I'm going to talk to you about the effects of decitabine in lung monocyte macrophages dynamics and how it affects the phenotype in the lung graft. I have no conflicts of interest to disclose. And we know that preventive treatment for acute transplant rejection focus on the effect on the adaptive immune response. And we previously showed in one of our animal models the protective effect of decitabine involving the adaptive immune response. However, the role of innate immune cells in acute rejection is not entirely understood. And from now on, I will be talking about decitabine. I will mention it as DAC, so we don't get confused a little bit. So our objective was to determine the role of both host and donor innate immune cells in DAC's anti-rejection effect. And to do so, we used an orthotopic murine transplant model where we transplanted the left lung of a biopsy donor to a B6 mouse. To differentiate between donors and host cells, we use CD45.1 B6 mice as the recipients and the biopsy as the donor. Normally, mice, the CD45 marker in mice is known as CD45.2. We did the transplant and we followed the mice for 10 days. And during day 3, 4, 5, and 7, we administered either DAC or DMSO, that was the vehicle where we dissolved DAC. At day 3 and at day 10, we harvested the tissues. And the reason that we harvested the tissue at day 3 is to see how the tissue looked at the same day that we started the treatment. We use histopathology and flow cytometry to do the analysis of the injury. And now here we have the gross anatomy and the histologic sections of our lungs. On the far right of these slides, we can see the specimens of the DMSO and the DAC groups at day 10. In the DAC group, we can see that the transplanted look much more aerated with less alveolar, peribronchial, and perivascular infiltration compared to DMSO. which we can see that's basically no irradiated areas. Now, if we compare this, now if we compare this to day 3, which is the starting point, even to a naive donor lung, we can see that the DAC treated group at day 10 looks very similar to the day 3. It has a little bit more of cell infiltration, but in general, we can see that the morphology is very similar. So at least histologically, we know that decitabine helps to attenuate the rejection. Now, from a cellular standpoint, we can see also a big difference. Now, let's focus first on the total number of cells in suspension and also in the live cells. So here in the histograms, I'm showing in gray the total number of cells, and in white, the total number of live cells. We can see that on day 10, the DMSO group compared to the DAC group had a higher number of total cells, but a extremely lower number of live cells compared to DAC. If we go to the day 3 treatment, which is the starting point, we can see that both DAC and the starting point are very similar in both absolute numbers and live cells. Now, if we go to the origin of the cells, if they are CD45.1 or CD45.2, which are the host cells, for example, we can see that in the DMSO graft, basically all the cells are coming from the host. They're infiltrating, and it's almost 0% of the donor cells. But if we see in the DAC group, we also have a higher percentage of host cells that are infiltrating the graft, but at least we have 4% of those donor cells that are still present in the graft. Now, if we focus on the monocyte macrophage population, now this is a very interesting story. Before starting, I want to walk you through the slides or the graph because it might be a little bit confusing. So on day 0, we have the donor naive lungs or the regular number of cells in a donor lung. Day 3, as I said, is the starting point, and day 10 is our final treatment point. So here I'm showing you the host-derived monocyte macrophages seen in the allograft. Looking to the alveolar macrophages, we can see that while a naive B6 mouse has about 10 to the 6 cells, there were essentially no host cells in the allograft by day 3. And we do see some infiltration of hosts into the alveolar space by day 10, but they did not come close to normal numbers. Also, the macrophages from the DMSO group had a pro-inflammatory phenotype compared to that group that showed an anti-inflammatory phenotype. These were the markers that we selected as pro-inflammatory, the TNF-alpha as an anti-inflammatory, CD36, CD206, and a proliferation marker, Ki67. Now, if we go to the interstitial macrophages, we see that normal B6 have about 10 to the 5 cells, but by day 3 and the allograft, the infiltration came up to higher to 10 to the 6 cells. And we can see that at day 10, the DMSO group, which is the red one, had a higher infiltration of interstitial macrophages and also were pro-inflammatory compared to DAC that were anti-inflammatory. If we go to the LY60 negative monocyte macrophages, which are historically known as anti-inflammatory monocyte-derived macrophages that are entering the graph, we can see that by day 10, that treated mice had a preserved number of cells compared to the DMSO group that basically had non-LY60 negative monocyte macrophage-derived monocytes. And on the LY60 positive monocyte-derived macrophages, which are pro-inflammatory, we can see that even though at day 3 the infiltration was very high by day 10 in both group DAC and DMSO. The infiltration or the numbers were close to 0. If we see the donor cells, we can see that in general, in the DMSO group, in all the cell types that we're looking, they were nearly 0. The interesting part is that the donor-derived alveolar macrophages were basically recovered at day 10 compared to day 3, and they were all with an anti-inflammatory phenotype. Even though that the interstitial macrophages also increased in the DAC group, it was not near 0 compared to the DMSO group, and it was lower than day 3, also with an anti-inflammatory phenotype. Regarding the LY6E negative macrophage monocyte population, this was maintained over time. And with the LY6E positive population, also in the DAC group was near 0. With this, we can conclude that the treatment of host mice with DAC after transplantation reduced the loss of resident monocyte macrophage populations in the allograft and promoted the maintenance of recruited macrophages from the host with an anti-inflammatory phenotype. These findings suggest that monocyte macrophages may also play a role in the protective effect of DAC on the long graft acute rejection. Thank you very much.
Right. Thank you, Dr. Daccarett. Like we have, maybe 2 minutes for questions, comments. May I ask our panelists for quick questions and comments?
Could I get a clarifying point for one thing? First of all, were these BAL cells or digests that you did from the lungs?
Were digested tissue completely, including, we didn't separate the tissue from BAL or lungs.
Oh, but did you separate the donor allograft versus the host ones, or they were all together?
No, they were all together in the same tissue. And the way that we differentiated them was with the CD45.1 marker that was with the host, and we did it through flow cytometry.
Okay, so you took both lungs that were the endogenous lung and the transplanted lung, and you digested those together.
No, so we took the graft, and the graft had the infiltrating cells that went into the graft from the host. So we only digested the graft, and that graft had both the cells from the donor and from the host that were infiltrating the graft.
Thank you. So you didn't digest the non-transplanted lung?
Yeah, we did. We also did that. I'm not showing that data here because that's my presentation for the next ATS to see the effect of the transplant in the native lung of the host. So, but yeah,
that was my question.
Okay,
sorry, that's okay. Now we
know
know that's coming up. That'll be exciting. Sorry.
Okay.
Okay, looking forward to the next ATS. All right. So let me introduce the next speaker. The next
Can I
up is
ask one more question? What is the
real quick one? Sorry.
Oh, never mind. Mine's not quick. I'll ask you at the end if we have time.
Okay.
Yes. Appreciate that. So next up is Dr. Daniel Cook, MD, PhD at University of Iowa. The abstract title is CFTR prevents type 2 inflammation by reducing IL-4 signaling in Th2 cells.
Sorry about that. I was trying to unmute myself. I thank you all for giving me the opportunity to speak with you, and I'm excited to be able to share some of the studies that went into the abstract, and happy to get any feedback or thoughts. But I study cystic fibrosis, which is a common genetic disease due to mutations in a chloride bicarbonate channel known as CFTR. And loss of this function leads to a multi-organ disease, but of most interest to me in my studies are the airway disease that occurs when that chloride bicarbonate channel is lost. And this is characterized by recurrent inflammation and infection, mucus accumulation, airflow obstruction. But one aspect that often goes underappreciated is contributions of type 2 inflammation to CF airway disease. And this is probably best studied in a comorbidity known as allergic bronchopulmonary aspergillosis, which is termed ABPA. This is a Th2 mediated and IgE mediated hypersensitivity reaction to the aspergillus, and occurs in a pretty high prevalence within the CF population, ranging anywhere from 6 to 12%. Also equally interesting is that mutations in CFTR have been implicated in non-CF ABPA. That's to say that if an individual is diagnosed with ABPA, they have an odds ratio of about 10 of being a carrier for CFTR mutation, suggesting that not only is ABPA enriched for in the CF population, but the hypersensitivity reaction is also enriched for in individuals that harbor CFTR mutations without developing CF. And this is true for other type 2 inflammatory diseases, including asthma and sinusitis as well. So the etiology of type 2 inflammation is complex in CF and something that we're interested in studying. We first set out to ask whether or not type 2 inflammation had any impact on overall outcomes for CF individuals. And we did this by separating a large CF cohort developed from Vanderbilt's synthetic derivative, which is a de-identified medical database. In this cohort, we had a little over 200 or 480 individuals with diagnostic coding for CF and genotyping data to support that. We could separate them into individuals that had ever had elevations in type 2 inflammatory biomarkers, being either eosinophils greater than 300 or IgE greater than 100, which both of these labs are done on a yearly basis within this population. And we could separate those from individuals that never had elevations in either of those markers, as seen in these two lines. So the black line on this Kaplan-Meier curve being individuals with cystic fibrosis that have never had those elevations versus the blue line, in which they had. And on the y-axis, we have survival probability. On the x-axis, we have increasing age. You can see individuals that develop type 2 inflammatory biomarker elevations had worse survival compared to those that never did. And it was associated with a hazard ratio of 2.24 for death, suggesting that not only is type 2 inflammation enriched for in a subset of individuals with cystic fibrosis, but it also led to worse outcomes. And so this led me to the central hypothesis of my work, which is that loss of CFTR primes the immune system in advance of a type 2 stimulus, and this leads to a heightened type 2 inflammatory response when the stimulus is present. And so a lot of our work, we use murine models to help us study what's going on in cells relevant to type 2 inflammation. In this case, I'm going to be talking about Th2 cells. So to study the effect on Th2 cells, we start out with murine splenocytes and negatively enrich for a CD4 positive, CD62L high, CD44 low population, so a truly naive T cell. And this is just flow gating to show what these cells look like. Most important would be the last two gates on the bottom left, showing that we select for a CD3 high, CD4 high population, as well as a CD62L high, CD44 low, so truly naive population. We use these for our studies. We can take these cells and stimulate them to become Th2 cells using TCR ligation with anti-CD3 and anti-CD28, as well as the polarizing factor IL-4. We can stop the production of Th1 cells with the anti-interferon gamma. And we allow these to sit in culture for 3 days. And then at the end of 3 days, we can take the cells and analyze them for flow cytometry and take the secreted cytokines as well and measure the amount of IL-13 and IL-5 produced. And so here we're looking at, on the right, CD3 positive, CD45 positive cells. And in these gates, we're looking at IL-13 on the y-axis and CD4 on the x-axis. Not surprisingly, based on the enriched culture, nearly all of these cells are CD4 positive. But if we look at IL-13, we could see in that upward right gate in both genotypes that more IL-13 producing cells were present within our CFTR deficient cultures compared to our CFTR sufficient, suggesting that there was more IL-13 producing cells when CFTR is lost. We could do this over multiple donors and see that if we look on a per cell basis for the amount of IL-13 that's produced off these gates, we'll see that there's increased IL-13 MFI in our CFTR deficient CD4 positive T cells compared to our CFTR sufficient cells. Likewise, we can measure the amount of IL-5 and 13 by ELISA from the cellular supernate and see that loss of CFTR increases both IL-5 and IL-13 production by these cells, suggesting that loss of CFTR increases Th2 effector function. So then we wanted to know whether or not CFTR could have a cell intrinsic role in these cells. So we turn to our RT-PCR plot here that I'm showing you to look at whether or not CFTR was expressed at the transcript level. The left column is a ladder, and the middle column here is our CFTR sufficient CD4 positive RNA, in which with reverse transcription, we were able to see an amplification band indicative of CFTR expression in our CFTR plus or wild type CD4 positive T cells compared to our CFTR deficient cells. But we could also look at protein expression using immunofluorescence. And so these are CD4 positive T cells that were cultured. And we're staining for nuclear content in DAPI in blue, plasma membrane lectin in red. And then in our CFTR sufficient cells on the right, we also have CFTR staining in green. And so we could see within a subset of our CD4 positive T cells in our CFTR sufficient cultures that CFTR was present. And this was not seen in our CFTR deficient cells. So because we saw these effector function differences and a cell intrinsic role for CFTR, we hypothesized that loss of CFTR would increase Th2 mediated allergic inflammation. So to do this, we utilized Cree mice that were driven by the promoter CD4, as well as CFTR floxed mice to knock out CFTR selectively in T cells. And then we also used potent Th2 stimulus and alternary extract to drive an adaptive immune response using the challenge model that you see below, where over 2 weeks, we sensitized at the beginning of, over 3 days at the beginning of the challenge with either alternate extract or control PBS, and then challenge these mice at the end of 2 weeks over 2 consecutive days. And we can harvest the lungs from these mice to enumerate the cells that would be recruited into the BAL. And so here I'm showing you the eosinophil counts and lymphocyte counts from the BAL from these challenged mice. In the closed circles, you can see in both genotypes, mice that were treated with PBS control. Not surprisingly, when we treat with PBS, we don't get any eosinophil or lymphocyte recruitment in the BAL. If we treat with alternary extract, we get recruitment of eosinophils in both genotypes. But in the far right red open circles, you can see the mice that had CFTR selectively knocked out in the T cells, and we can see increased amounts of lymphocytic and eosinophilic recruitment within the BAL, suggesting that CFTR loss increases that type 2 inflammation. Lastly, we can also look at IL-5 and IL-13 within these mice and see likewise similar trends. And so that just brings me to the conclusions that type 2 inflammation is relevant in CF lung disease. We show here that it negatively regulates Th2 function. Wasn't able to go through the data today about IL-4 activity, but we think in part it's regulating the IL-4 GATA3 signaling axis. And with that, I can take any questions.
Thank you very much, Dr. Cook. This time, let me ask Dr. Sperling for comments or questions.
Yeah, no, it was great talk, great work. I was just wondering if you, how do I put this? It's a little bit of chicken and egg, right? You know, you're making the argument that CFTR is regulating, at least at the very last moment, is regulating IL-4 receptor signaling. But your in vitro work, you threw in a bunch of IL-4. You know, what happens if you don't put in any IL-4? Do you find that you can get more responses?
Yeah, and it's a unique model. Obviously, we can polarize these cells into a number of different subsets. The IL-4 itself obviously helps them become Th2 cells. Now, if we just provide TCR ligation with anti-CD3 and anti-CD28, the sort of natural progression is to become a Th1 cell. We will see in culture, just with TCR ligation, there's a higher percentage of Th2 cell differentiation within culture and our CFTR knockout cells, again, reflecting that. Now, that might be, you know, sort of that chicken or egg that as the cells become polarized, they're producing more IL-4 that's feeding into the system. Here I'm showing you that we can provide increasing levels of IL-4 in culture and at least see a dose response whereby there's increased level of GATA3 expression in IL-4, more so specifically in that little intermediate to lower range of IL-4 that we'd normally use in culture and polarization, suggesting that they're at least initially within these first 3 days primed to sort of hyper react to the IL-4.
This is good data. The last question is, do you have any evidence that just sitting around in your clean / microbiome film mouse facility that these mice are developing any Th2 response before you start? I know you started with naive cells, but
yeah, that's a great question. Obviously, these mice can vary by facility. We never saw that phenotype at Vanderbilt where, like, say, IgE level just at baseline was increased. It sort of trended there, very low levels. It'll be interesting to see as I'm bringing up the colony here at Iowa if we have any sort of phenotype that manifests that way.
thank you.
Thank you very much. Let me get to the next presenter. Dr. Kritika Khanna, PhD at the University of California, San Francisco. Dr. Khanna will talk about her abstract that titled High Airway TSLP Levels in Asthma Associated with More Severe Disease and Multiple Biomarkers of Type 2 Inflammation. Please go ahead and share your screen, Dr. Khanna.
Hi, everyone. Thank you so much for this opportunity to present our work at this platform today. I'll be talking about airway TSLP levels in asthma and how they're associated with type 2 inflammation. So TSLP sorry, I think my slides are not progressing. TSLP or thymic stromal lymphopoietin is an alarmin cytokine. And as the name suggests, it is released by cells when they're exposed to environmental insults like allergens, viruses, bacteria, or even mechanical stresses. And it is implicated in multiple diseases like asthma, atopic dermatitis, or chronic rhinosinusitis. Through in vitro models, it has been shown that TSLP has a role in type 2 inflammation. It can directly act on dendritic cells to lead to the differentiation of CD4 positive naive T cells into Th2 cells, or it can act on CD4 positive naive T cells to convert them to Th2 cells. It can also act on group 2 innate lymphoid cells to activate eosinophils and promote Th2 inflammation. And TSLP can also directly act on eosinophils to activate them. In addition, there are a couple of reports that also suggest the role of TSLP in non-type 2 inflammation. Under special cases, it has been shown that TSLP can act on dendritic cells to lead to the differentiation of CD4 positive naive T cells into Th17 cells. Now, while there is information about in vitro roles, there is still relatively limited information about the expression of TSLP in the airways of asthma patients. And it is not known how these airway TSLP levels relate to gene networks for type 2 or non-type 2 immune responses. Notably, there is a monoclonal antibody called tezepelumab, which blocks TSLP and has been shown to improve asthma control. So with this background information, we formulated 3 key questions for our study. First, are these airway TSLP levels increased in asthma? And if what are the clinical features of these asthma patients with high airway TSLP protein levels? And finally, if these TSLP protein levels associate with any specific immune responses in the airways. So coming to the first part, we utilized this electrochemiluminescence-based highly sensitive assay to measure TSLP in the sputum samples. In our initial validation, we see that as the sputum is diluted, the electrochemiluminescence reads also decreased. And when we spiked in recombinant TSLP in the sputum sample, we have a good recovery through this assay. For the study, we utilized the Severe Asthma Research Program, or SART-3, which is a US-based multi-center program. And the cohort is enriched for patients with severe asthma, and it is very well phenotyped. For this study, we used the baseline measures and visit samples. When we looked at the sputum samples, we see that patients with asthma had significantly higher levels of airway TSLP as compared to healthy controls. And when we used the upper 95th percentile of the TSLP levels in health to divide the asthma patients, we see that about 34% have high airway TSLP levels. Next, we wanted to look at the clinical features of asthma patients having these high airway TSLP levels. And what we find is that those with TSLP high asthma were older, predominantly male. They had more severe asthma based on the ERS ATS criteria. And they also had increased use of oral steroids and were prone to exacerbations. When we looked at lung function, we see that TSLP high asthma was associated with lower lung function, as can be seen here by lower FEV1 and FVC. We next looked at markers of type 2 inflammation and found that those with TSLP high asthma were associated with increased nitric oxide in their exhaled breath and increased numbers of eosinophils in the blood and sputum. We also looked at sputum eosinophil peroxidase, which is this enzyme secreted by activated eosinophils and was very recently shown by the lab to be a better marker for airway eosinophilic inflammation. What we find is there is a very strong association of airway TSLP levels with airway EPX levels. When we divided the patient cohort into EPX high and EPX low asthma, we see that those with EPX high asthma had significantly higher level of sputum TSLP. And about 90% of the TSLP high asthma patients were also EPX high. We next looked at markers of non-type 2 inflammation and found that neutrophils in the blood or in the sputum were not significantly different between the TSLP high and low subgroups. We also looked at markers of metabolic asthma and found that airway TSLP levels did not associate with BMI. There was some association with history of hypertension and diabetes, but it was not significant after adjusting for age. We did not see any association with levels of insulin resistance. We did see some association of airway TSLP levels with plasma IL-6 levels, but it was only modestly significant after adjusting for age. So overall, it was just we just see a weak association with plasma IL-6 levels, but not with any of the other markers of metabolic asthma. So finally, we wanted to look at the airway protein TSLP levels and if they are associating with any specific immune responses, just to get a deeper understanding of what happens in the pathologic context. And for this, we leveraged the sputum transcriptomic data, which was generated in Max Seibolt's lab over at National Jewish. What we had done is looked at gene networks, which are networks having co-expressing genes corresponding to either type 2 or type 1 or type 17 inflammation. And we tried to associate these, the expression of these gene networks with airway TSLP levels. What we find is that TSLP high asthma was very strongly associated with increased expression of type 1 gene network. When we looked at the expression of type 3 gene network, we did not see any difference between the two subgroups. For the expression of type 1 gene network, we see a modest increase in patients with TSLP high asthma as compared to TSLP low. We next divided our patient cohort into subgroups based on expression levels of type 1 or type 2 gene networks and found that those with either dual inflammation of type 1 and type 2 gene networks or type 2 high gene network expression had higher levels of sputum TSLP. So overall, we see that TSLP high asthma is associated most strongly with the type 2 gene network expression and only modestly with type 1 gene network expression. So to sum it up, we see that in the asthma patients, TSLP protein levels were increased in airway secretions. And this airway TSLP protein levels were most strongly associated with clinical markers of type 2 inflammation. With our transcriptomic data, we see that airway TSLP protein levels associate most strongly with gene expression of type 2 immune responses and only weakly with that of type 1 immune responses. With this, I would like to thank you all for your attention, and I'm happy to take any questions.
Very interesting. I'll ask maybe one question from the panelists.
I'll go. So this is very, very nice. I was wondering, even way back in the very beginning, you were talking about all the cells that respond to TSLP. And my guess is that some of these patients can be on anti-IL-5 or anti-IL-5 receptor therapeutics. And I was wondering if you had looked to see if there's any relationship between when you get rid of eosinophils and you don't get rid of eosinophils, what happens to TSLP levels? Whether you could kind of imagine them going up, right?
Yeah, actually, thank you. That's quite an interesting question. In this study, we only looked at the patients with which were in the baseline visits. So they were not treated with any of the biologics. And I have not done the study in a longitudinal way, so I don't have data for the visits wherein they were already on biologics. But it would be interesting to see what happens when they are on the inhibitors for IL-5 or even TSLP.
Thank you. Really nice work.
Thank you.
Thank you very much. We are just on time. Let me introduce the fourth presenter, Dr. Andrew Moore at the Stanford University. His abstract title is 42 gene severe or mild SOM gene expression signature is conserved across viral and bacterial infections and is associated with differential response to steroids in septic shock.
Thank you all for giving me the chance to present today. As mentioned, my name is Andrew Moore, and I'm a postdoctoral fellow in the Rogers and Khatri labs at Stanford University. Today, I have the pleasure of presenting our work looking at the association of the 42 gene signature with severity in bacterial infections and its ability to differentiate patients with responses to septic shock, or with response to steroids in septic shock. I have no disclosures. So as sort of background for this, I think we all know that sepsis is a very heterogeneous disorder. It's a syndrome defined by sort of non-specific clinical and laboratory markers. And as a result of that heterogeneity, we really haven't come across any successful treatments for it. So there's this strong need for any sort of biomarker-based signatures to be able to identify subgroups of patients with differential sort of prognostic and treatment significance. And so today I hope to show you that we've identified a host response signature that's not only present in viral, but also present in bacterial infections, and then demonstrate its association with differential treatment response to steroids. So this work is based on a 2021 paper from one of my lab mates, Hong Zheng, who did an extensive multi-cohort analysis of peripheral blood gene expression in viral illnesses and identified 4 gene expression modules that were associated with severity. These 4 modules included 2 detrimental modules, which are associated with higher severity in viral illnesses. So on the x-axis here, what you can see is the severity of the illness, and then on the y-axis, you can see the module score. And for module 1 and module 2, you can see as severity goes up, these scores increase as well, showing their association with severity. And then she further found 2 protective modules. So module 3, which is really only enriched in these sort of mild cases of viral infections, and then module 4, which you can see decreases as severity increases. And what she found is that these modules, so module 1 and 2, are predominantly sort of innate activation, predominantly immature neutrophils. And then module 3 is sort of this monocyte signature, and then module 4 is predominantly these protective lymphocyte signatures. Together, these gene modules can combine to create the severe or mild score, which is 2 detrimental modules divided by the protective modules. And as you can see across both the discovery and the validation cohorts, they're highly associated with severity. So this brings us to our first question. So this gene expression signature is broadly conserved across viral infections, but the next logical question is this signature also seen in bacterial infections? So we gathered a total of 30 data sets, inclusive of bacterial and viral infections, across the full spectrum of severity, as you can see here, curating a total of over 3,000 samples. We then co-normalized these samples and evaluated these scores across viral and bacterial infections. And importantly, we found that this gene expression host response signature was associated with severity across all bacterial infections interrogated. So here we're just looking at bacterial infections. And on the x-axis, again, you can see severity, and on the y-axis, this SOM score. And you can see SOM score increases as severity increases. Now, I didn't show the data just for sake of time, but as you'd expect, detrimental scores increased, protective scores decreased. And this was present across all the different bacteria, even when we disaggregated the data. And then here you can see the AUROC for predicting severe from non-severe bacterial infections. And you can see that it really is able to differentiate severe from non-severe infections with relatively good discrimination. So where the rubber really hits the road, though, is can we use these gene expression signatures to identify subgroups of patients who benefit from or may be harmed by therapies? So I know that many of you are already well aware of the work done out of the Knight group, looking at heterogeneity of treatment effect based on the transcriptomic signature in the VANISH cohort. But as a reminder, the VANISH cohort was a factorial, double-blind, randomized controlled trial in the UK. It enrolled patients with septic shock within 6 hours of developing hypotension requiring vasopressors to first vasopressin and norepinephrine, followed by hydrocortisone or placebo. So what we initially did was we looked at the effect of the SOM score and steroid treatment on mortality. So what we're looking at here is an interaction plot. So what we did is logistic regression for 28-day mortality, looking at the interaction between steroids and SOM score, adjusting for age, sex, and Apache 2. So what we're showing here is the marginal effect that steroid treatment in pink has on mortality on the y-axis relative to the SOM score, which is on the x-axis as that SOM score changes. And what you can see is that at lower SOM scores, so these patients who are relatively protected, receiving steroids resulted in a higher mortality. Whereas you move towards higher SOM scores, this mortality decrement was reduced and potentially reversed. Now, what I really like about the SOM score is that you can then split the immune responses into its detrimental and protective responses. So the next question really becomes, what was the driving effect of steroids? So on the left, you can see the same plot, but looking at detrimental host response. And what you can see is that there really isn't a differential response by detrimental score. However, on the right, when you look at protective score, you can see that this treatment difference was predominantly driven by patients with this high protective score. So patients with a good, appropriate immune response who experienced higher mortality if they received steroid treatment. So then the other question we had is whether we could use a cutoff to determine who might benefit from steroids versus who might be harmed. So we evaluated high versus low protective score just based on a median cutoff seen here on the x-axis, with high protective score on the right and then low protective score on the left. And then we looked at mortality on the y-axis, separated by steroids in pink versus no steroids in gray. And you can see that patients with this high protective score at baseline who received steroids did indeed have a significantly higher mortality at 45% relative to 15% in those who did not receive steroids, suggesting that we may see a differential response in steroids in septic shock patients depending on their protective immune responses. So in conclusion, the severe or mild gene expression host response signature is conserved across bacterial and viral infections and may be associated with differential treatment response to immunomodulation. I'd like to thank my mentors, Angela Rogers and Pravesh Khatri, my co-fellows and lab members, Hong Zhang and Anant Ganesan in particular, and then the Stanford Pulmonary T32. I'd also like to make a little plug, which is that this work was part of a much larger body of work that's under review and available as a preprint. So if you're interested in this, as well as some other immune modulating therapies that we've looked at, like anakinra, and you have the time, we'd love for you to check it out. Thanks so much, and happy to take any questions.
Thank you, Dr. Moore. Yep.
Oh,
Go ahead.
I'm really surprised that you see the same effect for viral and bacterial. Being an immunologist, I think there's a very different immune response that happens with one versus the other. So can you kind of speculate on why you think that this protective response really was kind of agnostic of what the infectious agent was?
Yeah, and so We go into this a little bit in depth in the preprint, but we do definitely see differences between viral and bacterial infections. And it's known that, like, based on gene expression, we can actually differentiate bacterial and viral infections just based on gene expression. It does seem like once someone gets to a severe enough infection, that sort of like dysregulation that happens tends to be a little bit more conserved, with just this innate immune response that just goes out of control. And then in particular, it sort of downregulates this protective NK and CD8 and CD4 T cell response. And so that seems to be the sort of conserved portion of it, whereas the initial activation portion of it, I think, is very different between viruses and bacteria.
Kind of the difference with the innate and the adaptive is kind of moving forward, that the innate responds to everything, may be slightly different based on the virus versus the bacteria, but it seems like the adaptive is a less specific, I guess, in some ways.
Yeah. Yeah, and I mean, there's so much to do with it. Like, I mean, like even within bacteria, I'm sure there's like an appropriate immune response to your bacterial insult and an inappropriate response. And we still have a lot of work to do there as well.
Very interesting.
Thank you so much. Let me introduce our final speaker today, Dr. Shaun Pienkos, and also at Stanford University. He's going to talk about the abstract titled Patient-Reported Symptoms Do Not Correlate with Immune Profiling in Multi-Omics Analysis of Post-Acute Sequelae of COVID-19.
All right. Well, thank you, everyone, for joining today. Again, my name is Shaun Pienkos. I'm a postdoc and physician at Stanford. And as Tetsuro mentioned, this was the abstract title that I submitted in late 2023. A lot has changed since then, and we've actually published the work based on this data set. And things look a little bit different now over a year later. And the published work is titled Sex Differences in Immune Correlates of Long COVID Development, Symptoms, Persistence, and Resolution. And so I'm going to describe some of our findings in that paper today. I have no financial disclosures to share. And as a brief overview, I'm going to describe a little bit about the study cohort and questions we aim to answer. I'm going to review one example of immunologic sex differences we observed during acute COVID infection, which predicted development of long COVID versus normal recovery. And then discuss increased interleukin-4 findings in T cells of individuals of both sexes with long COVID persisting to 12 months, and also talk about a corresponding transcriptional difference. So first, I'll share that we were hoping to answer two questions with this data. First was, what features of acute COVID-19 predict recovery versus long COVID? And second is, what immune features correlate with persistent long COVID versus recovery between 3 and 12 months? And we chose these time points of 3 and 12 months somewhat arbitrarily, but that's because in the bottom panel, you can see this is when we had blood samples available. So we collected samples during acute infection and then at 3 and 12 months after acute infection. During acute infection, we had samples from 21 patients, 15 of whom went on to develop long COVID and 6 of whom recovered by 3-month follow-up. And then we had paired blood samples from 23 patients, about half of whom had long COVID at both 3 and 12 months, and half of whom had long COVID at 3 months, but recovered by 12 months. And then moving to the top right, just showing what we did with these samples, we did a multi-omic analysis, first doing plasma proteomics with the Olink platform, and then we also analyzed peripheral blood mononuclear cells, performing CyTOF, which is a single-cell proteomic method, as well as single-cell RNA sequencing. So our cohort overall was mostly mild infections. There were just a few who were intubated or and/or hospitalized. But as far as symptoms reported after the infection, during our 3 and 12-month follow-ups, we had a wide range of symptoms, including cardiac, pulmonary, neurologic, and gastrointestinal, as reported in other cohorts. As you can see here, we've split the bar plot by sex, which will make more sense in a moment, but did want to point out that there were not symptom differences between males and females in this cohort. And then lastly, I wanted to mention that our definition of long COVID, since this has been a bit of a moving target, is consistent with the current WHO definition, which is symptoms persisting greater than 3 months after acute infection, which are not explained by another disease process. So to start, I'm showing some of our single-cell transcriptomics data, and we used something called blood transcription modules, which are just gene sets which have proven useful in analyzing vaccine and virus cohorts in the past. And on this heat map, you can see that gene sets are on the y-axis and cell types on the x-axis. And red boxes indicate gene sets which are upregulated in those who go on to develop long COVID, while blue boxes are gene sets which are downregulated in those who develop long COVID. And here we're looking at samples acquired during acute infection. The main takeaway on this slide is that there was a chromosome Y-linked module which was downregulated across many immune cell types in our cohort. And this is a gene set of 17 sex-linked genes. And so because of this suggestion of sex-specific immune differences, we went on to analyze the sexes separately for much of our analysis. So here I'm showing a similar heat map with males in the left panel and females on the right panel. And what I'd like to draw your attention to here is that there was a particular cell type which seemed markedly different between the sexes, and that was proliferating NK cells or natural killer cells. There were a lot of gene modules which had different expression levels between those who developed long COVID and those who recovered by 3 months in males, while this difference was much less pronounced in females. Because of these sex-specific differences, we went on to examine inferred cell-cell communication, differences between males who developed long COVID at 3 months versus the males who recovered by the 3-month time point. And on these plots, what I'm showing is ligand receptor pairs, where the ligand is at the base of the arrow and receptors are at the tip of the arrow. And the tip of the arrow also corresponds to receiver cells for signal that would be sent by proliferating natural killer cells. And what I've highlighted here is TGF-beta1 expression from natural killer cells communicating to many other lymphocyte cell subsets in patients that had long COVID at 3 months versus those who recovered. There was no prioritization of TGF-beta1 signaling. Because of this sex-specific difference in TGF-beta1 in natural killer cells, we also examined TGF-beta1 across other cell types. And in the left panel here, I'm showing TGF-beta1 expression in single-cell RNA sequencing data, with bars to the right indicating higher expression in those who had long COVID versus bars to the left being lower expression in those who had long COVID. And again, you can see sex-specific differences with males showing increased TGF-beta1 expression across almost all cell types during acute infection, whereas females who developed long COVID had reduced TGF-beta1 expression during acute infection if they went on to develop long COVID. In the middle here, you can see that we also had a plasma proteomic correlate of this finding. LAP TGF-beta1 is a TGF-beta precursor protein, which was in our proteomic panel. And we found that males who went on to develop long COVID had higher LAP TGF-beta1 during acute infection, but this pattern was not seen in females. Next, I'll move on to one of our most interesting findings from our 3 and 12-month samples. And here again, we were trying to answer whether there were immune correlates of persistent long COVID going out to 12 months versus those who recovered by 12 months. And what we found was in our CyTOF or single-cell proteomic data that when we measured interleukin-4 in T cells at both 3 and 12 months, we found that those who had long COVID persisting to 12 months, as indicated by the red boxes and dots, had higher interleukin-4 in both CD4 positive and CD8 positive T cells at both time points. And that was in comparison, again, to those who recovered by 12 months after acute infection. We then turned to our single-cell transcriptomic data to look for correlates of this increased IL-4 or Th2 marker. And we found that a transcription factor called ETS1 was reduced in both males and females across basically all lymphocyte cell subsets at both 3 and 12 months for those that had persisting long COVID to 12 months. ETS1 is important in proliferation and apoptosis for lymphocytes, but also Th1 and Th2 balance. And it's been studied in the context of autoimmunity most extensively in both mouse and human models, and found that ETS1 deficiency is associated with more autoimmune activity, as well as higher Th2 markers, including IL-4, IL-5, and IL-13. So we found this particularly interesting since, as you know, pulmonologists, I think we're all aware of highly effective Th2 modulators in our eosinophilic airways diseases. And so existing therapies may be able to address at least a subset of patients with long COVID. So with that, I'll just wrap up with conclusions. So in our cohort, we found that sex differences underscored the immune dysregulation found to distinguish those that progressed to long COVID. We found that upregulated TGF-beta signaling in males was associated with long COVID development at 3 months. And then we saw that long COVID persisting to 12 months after acute infection was associated with increased interleukin-4 in T cells and a corresponding reduction in ETS1 expression across lymphocyte cell subsets. I provided a QR code in case any of you are interested in looking at our recent publication. And thank you for the opportunity today, and happy to answer any questions.
Thank you very much, and congratulations on publication. Maybe I'll ask for one question for Dr. Pienkos. Any question?
Megan, you don't have one? Does anybody have one?
Well, I guess I had one question. I mean,
Yeah.
were these patients, did they have asthma? Did they have a Th2 like disease beforehand? Did that make them more predisposed? I mean, were there any differences on what they had before compared to kind of some of these Th2 skewing responses that you observed later?
Yeah, it's a great question. We only had one of the total 45 patients that we included in this manuscript were on inhaled corticosteroids before enrolling. And so as far as patient reported as well as kind of medication records, there was not a high prevalence of asthma before. We, in our cohort, didn't have, I think, the sample size to say much about respiratory symptoms in particular being more prevalent with higher interleukin-4 or this ETS1 transcription factor. But in another cohort that did plasma proteomics, we did see that interleukin-4 was associated with a respiratory long COVID phenotype, and they had hundreds of patients, so we were able to kind of see that pattern more clearly.
Was it just IL-4, or what about IL-5 and 13? Were they up too?
Yeah, so in our cohort, we had 4, 5, 13, and 33, and I didn't show that in our, this was in our plasma proteomics. They were all increased, but the only significant finding was IL-33 in the, higher in the long COVID group compared to the recovered group.
And there was a way back in the beginning of all of this, Akiko published a paper where she defined a group of people who are getting big eosinophilic responses during the acute inflammation in the blood. And I was just wondering if you had any data from the acute that would predict this phenotype.
Yeah, that's a good question. I don't think that we did really have any findings in the acute samples that would predict Th2 inflammation. Since you mentioned Akiko, she kind of has actually replicated both the increased IL-4 correlating with long COVID, as well as TGF beta increase in males in some of her samples. So, I wanted to highlight those samples or findings because I think this is a difficult area and little has been replicated, but we're obviously happy to report some things in our data set were seen in others.
I actually asked her the same question, whether they were correlated with the eosinophils in the beginning, and she didn't have an answer either. So,
okay.
Right.
Not just so I'm very consistent.
Yeah.
Thank you all for awesome presentation. So we are coming to the end. But maybe at the end, I will ask each of the panelists to, for a quick comment. Dr. Sperling.
Oh, from us? I thought it was a great session. I'm sorry more people didn't come, because I'm glad you guys get to hear each other though, because it's really all the way from asthma, TSLP to sepsis. We covered it all, and transplant too. So it was very, very interesting number of presentations. I thank you all.
I'm just really excited that people are still finding new things about immunology. I mean, these cells have been around forever, and look at all the really cool things that we're still understanding, how they react in a human, new animal models, understanding CF and how this could predispose to different diseases, understanding the different systemic responses that we get to sepsis or to long COVID. I mean, there's just so much more to learn. And I think the lung is, as an immunologist, I've always said the lung was such a cool place. And now I think since COVID happens, everybody believes me that everything we breathe in gets into our lungs. And so having that robust immune response is so important. And I'm so excited about the work that you all have done. I hope you continue to do some great work, and we continue to see you at ATS.
Thank you very much. All right, so I would like to thank all of our speakers, panelists, as well as all of you that joined. And thank you to AII Assembly for sponsorship of this webinar. As a reminder, the recording of this webinar should be posted in about a week on the website. And there will be part 2 this Thursday, 1 p.m. EST. So please feel free to join that. And if you have suggestions for future speakers or topics, please consider contacting our ATS Assembly representative. And with that, we'll conclude our webinar. Thank you all again, and have a great evening.
And ATS registration open today. So hopefully everybody's going to be registering, and we can see more of this exciting work that has already been shown.
Thank you.
Thank you. That's it.
Thank you, everyone.
Thank you.